Diagnosis: virus isolation is often necessary to fully characterize an influenza virus outbreak in poultry. The virus is typically cultured in embryonated chicken eggs, but it can also be grown in several cell tissue culture systems. Virus is typically first tested for ability to heme-agglutinate chicken red blood cells. If the virus heme-agglutinate, it must be differentiated from other heme-agglutinating virus, including Newcastle disease virus, and typically is confirmed by an agar-gel immunodiffusion test. The standard pathotyping test for influenza is to inoculate 8 specific pathogen-free chicken aged from 4 to 6 weeks of age by intravenous inoculation and observe for death of birds within 10 days post-inoculation. If 75% or more of the birds die within 10 days, the virus is considered highly pathogenic. Serology can also be used to identify flocks that have been exposed to avian influenza virus. The most common tests are the agar-gel immunodiffusion test and the commercial ELISA test. The agar-gel immunodiffusion test detects antibody to both the nucleoprotein and matrix 1 protein that are conserved for all types of influenza virus. The commercial ELISA test detects only antibody to the nucleoprotein. Both tests are commonly used in diagnostic laboratories and can usually detect infection in a flock within a week of initial infection. Serum sample can also be used to determine the heme-agglutination and neuraminidase subtypes of the virus by using the heme-agglutination and neuraminidase inhibition tests. Both tests require testing the sample against a bank of reagents for all 16 heme-agglutination and 9 neuraminidase subtypes, and therefore it's only commonly done at large regional or national reference centers. AI outbreak include quarantine of the infection. If we need to control the AI outbreak, include quarantine of the infected flock, a quarantine zone around the infected flocks. Secondly, increase biosecurity by greatly restricting access of personnel and equipment to and from farms in the quarantine zone. Third, increased surveillance on the surrounding farms to monitor for evidence of spread of influenza infection for outbreak of highly pathogenic avian influenza. Infected flocks are depopulated, often with disposal of the birds by burning or burial on the farm. For low pathogenic avian influenza outbreaks, the outcome of the infected flocks varies, but often the infected birds are allowed to recover from the infection and then are marketed with special precautions. 2 types of vaccine are available for use in poultry: a killed whole virus adjuvanted vaccine and a live vectored recombinant vaccine. The killed whole virus recombinant vaccine uses embryonated egg-grown virus that is adjuvanted with an oil emulsion and administered either intramuscularly or subcutaneously. The recombinant vaccine are vaccine using a fowlpox virus vaccine strain or the herpes virus turkey as a vector, in which the heme-agglutination gene from influenza virus is inserted. When the vaccine is administered, the fowlpox virus or the herpes virus will produce both fowlpox protein or herpes protein and influenza heme-agglutination protein that the host mounts an immune response to.